Hello students, we are our next part, that is fundamental unit of life, part 2. I hope you enjoy the first video. So we are going to start our this chapter. Look at it. So students, in the previous video we are study about the prokaryotic cell in the detail. Now in this video we are going to study about the eukaryotic cell. As well, we discussed in our previous video about the cell discovery, cell theory, types of cell, prokaryotic cell in complete detail. Here, we will learn about eukaryotic cell in detail. So, we already discussed about eukaryotic cell in small way, that is, eukaryotic cell are the such cells which have true nucleus. True nucleus means their genetic material, that is DNA, जिसकी definition या you can say जिसकी हम full form already कर चुके हैं, that is deoxyribonucleic acid, is surrounded by nuclear membrane which is absent in the prokaryotic cell. Such organisms which made up by eukaryotic cells are called as eukaryotic organisms. Eukaryotic organism can be unicellular or multicellular. Means they can be a single cell organism or they can be multicellular organism. Protista is the kingdom of unicellular eukaryotic organisms. Protista जो kingdom है which is divided under the classification is a complete kingdom of unicellular eukaryotic organisms. Example, that is amoeba, fungi, plant, animals. They are the three kingdoms which is the kingdom of multicellular eukaryotic organisms. Now we are going to study about the features of a eukaryotic cell. Now the first feature of eukaryotic cell, that all eukaryotic cell have the nuclear membrane. The second one is they have membrane bound organelles that is absent in the prokaryotic cell except one, that is ribosomes. That is the only organelle which is present in prokaryotic cell. They have 80S ribosomes which is made up by 60S and 40S. 60S is the larger unit and 40S is the smaller unit. Some of the eukaryotic organisms have sulfur. As usual, we know that the prokaryotic cell also having the cell wall, but what is the difference between both of them? We discuss it. In fungi, cell wall is made up of chitin. In plant, cell wall is made up of cellulose, and in animal cell there is no cell wall. So here is the two substances which made the cell wall, that is chitin and cellulose. But in case of prokaryotic cell, that the cell wall is made up by peptidoglycan. Now we are going to discuss about the difference between prokaryotic cell and eukaryotic cell. There we go. This one side represents the character of prokaryotic cell and the other side represents the eukaryotic cell characters. The first character of prokaryotic cell, true nucleus is absent. We already discussed about it, that is nuclear membrane is absent and the DNA, or you can say the single chromosome, is present in the cytoplasm. On the other hand, in eukaryotic cells, true nucleus is present, means that is surrounded by the nuclear membrane. In prokaryotic character, the second character is cell wall is made up of peptidoglycan, and in eukaryotic cell we already discussed, that is in fungi the cell wall is made up of chitin, and in plant the cell wall is made up of cellulose. They have small size. They ranges from 1 to 100 micrometer in prokaryotic cell. On the other hand, the size is large and their ranges is from 10 micrometer to 1000 micrometer in eukaryotic cells. In prokaryotic cell, the ribosomes are made up by two subunits, that is 50 and 30, that is combines and forms 70 S unit. As we discussed about, S represent the Svedberg unit, that is the centrifugation coefficient. On the other hand, in eukaryotic cells, the ribosomes are 80S, that is made up by two subunits 60S and 40S. In case of prokaryotic cell, respiration definition is breakdown of the food and release of energy is done by the plasma membrane. Because there is no cell organelle, the function is performed by the plasma membrane. On the other hand, in eukaryotic cells, the respiration process is done by the mitochondria, which is a membrane-bound cell organelle. All cell organelle absent except ribosomes, as I already told to you. In case of eukaryotic cell, all cell organelle are present. In prokaryotic cell, they have single chromosome and that is present in the cytoplasm. But in case of eukaryotic cell, there is the presence of many chromosomes and they present in the nucleus. Now there is the difference on the basis of their picture. Here we see that prokaryotic cell already discussed, and there is, on the other hand, there is the presence of the eukaryotic cell, which represent the all membrane bound organelle that is present inside the cell. Now we are already finished this, that is difference between prokaryotic cell and eukaryotic cell. I hope you all get about it. Now we are going to discuss about the difference between animal cell and plant cell. Here we go, that is one side it represent the animal cell and the other side represent the plant cell. The first character of animal cell, that is animal cell don't have cell wall. We already discussed about it. And on the other hand, the plant cell have the cell wall that is made up by cellulose. In animal cell, there is many number and small size vacuoles are present, and in plant cell, it contains single and large size vacuole. We will discuss vacuoles in this video on later. That is, the next is endoplasmic reticulum. In case of animal cell, that is directly attached to the nucleus. On the other hand, in plant cell, they are present near the nucleus. In animal cell, don't have plastids, which is required for the photosynthesis, as usually we know about it. Plant cell have the plastid to carry out photosynthesis, and the animal cell have centrioles, which is absent in the plant cell. This is used for the cell division. Glycogen is stored food material in animals, and starch is the stored food material in plants. Number of mitochondria is higher in animal cell and number of mitochondria is less in plant cell, because due to presence of the plastid they can synthesize the energy by their own using of sunlight. And I hope you clear about it. Now we move further topic. This is the structure of plant cell as well as the animal cell. You can see each and every structure easily overhead, that is in center both of them having the same nucleus, and in case of plant cell the endoplasmic reticulum scattered, but in case of animal cell they are just close to the nucleus. Mitochondria number is large in case of the animal cell but less in case of plant cell. Plastid are representing into the, you can say the chloroplast represents to the plant cell, which is absent in case of animal cell. Centrioles on the right hand, that is red in color, represents that cell is animal cell, which is absent into the plant cell. Now we move further. Now we are going to discuss about the cell organelles. So first of all we have to know about, that is, what is cell organelle. So we all about the cell. In cell there is two parts present. The first part is cytoplasm and the second part is cell organelle. So first of all we are going to discuss about the cytoplasm. It is the liquid part present in the cell. It contains all minerals and salts etc. in it. It contains dissolved proteins and it provides space for all activities in cell. Now we are on the other hand cell organelle. They are the small size particle present inside a cell. They are small teeny particles actually. They are scattered in the cytoplasm. They help in activities performed in the cell. Example, mitochondria, endoplasmic reticulum, Golgi bodies, and so on. There are so many cell organelles. So first of all we are going to discuss about the endoplasmic reticulum, that is represented by ER also. Let's see. So endoplasmic reticulum, it is a network of two membranes which run parallel to one another and form a tube-like structure. This tube-like structure is known as tubule. Means to run two membranes running with one another and form a tube-like structure. This tube-like structure is tubules. Generally, ribosomes attach on it. It is generally actually. Types of endoplasmic reticulum. That is endoplasmic reticulum. It is of two types. That is, first is rough endoplasmic reticulum, that is known as RER, and the another one is smooth endoplasmic reticulum, that is SCR. Here is the difference between both of them. Rough endoplasmic reticulum, they are rough due to the attachment of ribosome on it. On the other hand, smooth endoplasmic reticulum is smooth due to absence of ribosomes on it. Second, they arrange near to the nucleus. They arrange away to the nucleus. They are help in synthesis and modification of protein. ये ज़्यादा important part है आप लोगों के लिए, because we know that ribosomes are responsible for the formation of protein and it attached to the RER. On the other hand, smooth endoplasmic reticulum helpful in the synthesis of lipid and steroids. Here is the picture. You see, in this picture it is clearly indicated that in the surrounding of the nucleus, that is bluish in color, it is clearly indicated that the endoplasmic reticulum is form a tube-like structures, actually tubule, and ribosomes attach over them and give them a rough appearance. It is present near to the nucleus, and that's why we easily differentiate them due to their appearance and their position, that is rough endoplasmic reticulum. On the other hand, the smooth endoplasmic reticulum is away from the nucleus and it not contain any ribosomes over it. So this is the picture. Now we discuss about the Golgi bodies and you can say Golgi apparatus. First time it was discovered by a scientist named that is Camillo Golgi in 1898. So they known as Golgi bodies on the name of the discoverer. They are also known as traffic police of the cell, as the traffic police controls the transporting means. As such, it controls and check the material transport during the cell, you can say within the cell or out of the cell. It checks the material for the transportation between the cells, as I already told to you. It packages the material for the transportation. Golgi bodies are made up of arrangement of membranous structures called cisternae. These cisternae arrange one over another. In the picture you easily see that cisternae arrange over one another. They are the sac-like structure actually. The space present between these cisternae is known as human. And you easily see that the transporting vesicle, that is on which the membrane is put by the Golgi body and pack them properly. It releases lysosomes. Actually, lysosomes we will discuss in this video after some time. It also releases secretory thickening substances for cell wall in plant. Such substances are known as cellulose and nanocellulose. Actually, these are the two substances which makes the cell wall of a mature plant cell. And these substances are further released by the Golgi bodies. It also releases the vesicles, which we discuss in after some time. Now the next one is lysosomes. They are also known as the suicidal bags. Suicidal bags because they burst them inside the cell and digest the complete cell. We will discuss in here. Look at it. It is single membranous structure contain hydrolytic enzyme. Hydrolytic enzymes are such enzymes which can digest protein, fats, carbohydrates, and you can say every substance that is present inside the cell. They present in saliva and they kill the bacteria present in the food. They also present in tear and sweat to kill the bacteria. They help in digestion of food in unicellular organism like in case of the amoeba. When injury occurs, they rupture on that place and prevent the entry of bacteria inside the body. It digests its own cell. But that is a particular condition when the cell is not functions, or you can say it become old, that digest it and release the nutrient from it. Now the next one is mitochondria. It is also known as the powerhouse of the cell. Powerhouse means which provide energy to the cell. This is the structure of the mitochondria. It is made up by double membranous, that is outer membrane and the inner membrane. Inner membrane is not smooth but outer membrane is smooth. It contains ribosomes, that is 70S. The space between these two membranes is known as intermembranous space. Here we see that the inner membrane is infold and form finger-like structure. This finger-like structure is known as cristae. This cristae having small projections over it. These small projection is known as oxysomes. And the space, total space that is present inside the inner membrane, that is known as matrix. In matrix, there is presence of ribosomes and mitochondrial DNA, that is its own DNA. So we discussed that mitochondria is cylindrical in shape. It made up of two membranes, outer and inner membrane. We already discussed that, and the space between both two is known as intermembranous space. Inner membrane infold and for finger-like structure called cristae. On cristae, small projections are present called oxysome, in which respiratory enzymes are present that helpful in the making of ATPs, that is adenosine triphosphate, that is a energy currency of our body. Mitochondria can also divide by on to form its many copies by the process fission. This is the structure again we are going to discuss, that is regarding the mitochondria. So this is the picture. The space present inside the inner membrane is known as matrix. We already discussed about this, in which all activities are performed. Mitochondria have its own DNA. DNA की full form that is deoxyribonucleic acid. It have its own ribosomes. So as we study that prokaryotic cell, that is already have the part which produces energy, that is plasma membrane and the ribosome, and they can survive because they have DNA, they have ribosomes and energy producing unit. That all character also present in case of mitochondria. So if we remove the mitochondria out of the cell, it can survive by its own. Such type of organelle is known as semi-autonomous organelle. So here the definition: it can survive after isolation from the cell. Such organelle is known as semi-autonomous organelle. Now we are going to discuss about the plastids. What are plastids? They are also known as the kitchen of the cell because kitchen is useful for making of the food. So plastid is also helpful in the formation of food, as we know that the plastid is present only in case of plant cell and absent in animal cell. They are helpful in the formation of food in plant cell. We already discussed this. Now this is the structure of plastid. This is also made up by two membranes, that is outer membrane and the inner membrane, and that is the space that is known as intermembranous space between these two. In inner space, you can say the space present inside the inner membrane, that space have particular name, that is known as stroma, which is present on our left hand. In stroma there is the many coil-like structure in the arranging form, that is known as granum, and this single coin is known as thylakoid. These granum attached to one another by the lamellae, and the thylakoid having a particular lumen of its own. Now we are going to discuss that is plastid is double membranous structure. that is outer and the inner membrane, and the space present between these membrane is called intermembranous space. Same here we are going to discuss regarding the plastid. Inside the inner membrane, the space is present. We already study about it. That is, we already discuss about it. That is, the space is known as stroma, in which all activities are takes place. In stroma, coin-like structure are present. This coin-like structure is known as thylakoid. They trap sunlight. Actually, this is the main function of the plastids. That is, they trap the sunlight and converted it into the chemical energy required for the photosynthesis. Thylakoid arrange over one another and form a structure that is known as grana, that is singular, and the granum is plural. These granum attach to one another by the membranous tube called stroma lamellae. Plastid have its own DNA and ribosome as such of the mitochondria. So, if it is removed from the cell, it can survive by its own. So, it is also semi-autonomous in nature. Now, we are going to discuss about types of plastids. So plastids are of mainly three types actually: chloroplast, the second is chromoplast, and the third is leucoplast. This is chloroplast, such plastid in which chlorophyll pigment is present, and we already about this is the green pigment. They are green in color. They trap the sunlight and help in photosynthesis. Now the chromoplast, as the word indicated, chromo means colorful. They are found in the colorful part of the plant, and the colorful part is flower in case of the plant. They give color to the plant for pollination. Pollination is the process of transfer of pollen grain. We will discuss further. Leucoplast, they found in the colorless area, and the colorless area mainly present inside the soil, means generally they store the food in case of plant. Now we are going to discuss about vacuoles. They are single membranous structures. They have different material in them. On the basis of type of material, they get their name. Their membrane is known as tonoplast. Actually, it is very important, the name of the membrane, that is tonoplast. So there is the different types of vacuoles. The first type of vacuole is food vacuole. As the name indicates, it is contain food in it. The second one is residual vacuole, and the third one is contractile vacuole. Here is the detail. Food vacuole, it contains food for the digestion. Residual, after digestion, undigested food remain in the vacuole. Such vacuole is known as residual. Here is the contractile vacuole. It helps in osmoregulation. The word osmoregulation means balancing of water level inside the body, and the excretion, that is removal of nitrogen waste. Here is the one term, that is osmosis. Actually, osmosis, it is the process by which water moves from higher concentration of water to lower concentration of water through semi-permeable membrane. Semi-permeable membrane, it is the type of membrane which allow only pass selected particles, not every particle. It is only allow the selected particles. This is the diagram we use for the osmosis. So in this picture, assume there is the two sides are present. Look here, this is A side and this is B side. Both of the sides having the same level of material. Look at the upper picture. The white particles represent to sugar and the black particle represent to water. As we see that inside A, there is presence of sugar with water, so the amount of water is less than the B. If we assume that side A having 80 ml of water and 20 grams sugar, then side B having completely water, that is 100 ml of water, so the water amount is higher in side B than the A, and it is separated by semi-permeable membrane. Then the concentration of water is high on side B. We are not counting sugar actually. So water is moved from higher concentration of water towards lower concentration of water, that is from B to side A, which is seen by the lower picture. So thank you students. So if you like this video, please like it and subscribe the channel. Thank you.